Pulldown assays, most commonly in the form of co-immunoprecipitation or co-IP, are a form of experiment that allow you to test if proteins or other molecules are interacting with one another. The way that you do this is by pulling out or removing from a mixture a protein and looking to see what that protein was interacting with. Most commonly with a Western blot, but sometimes you can also use mass spectrometry, which would be an IPMS. The way that you're going to pull out the protein is by using little beads that are attached to something that will bind to your protein specifically. In the case of a co-immunoprecipitation, those beads are attached to an antibody that was going to recognize a protein of interest called your bait. The bait is only one of the proteins of interest. It's the protein that you're going to be directly pulling out of the solution. And then you want to see, well, what is it actually bound to? The things that it's bound to are going to be called prey. So, for example, in the case of a co-immunoprecipitation, if we were to say maybe this protein is called poly, we would have an anti-poly antibody that was conjugated or covalently attached to a bead. And then we would isolate the beads, and by pulling out those beads, we're able to pull out the polyprotein that was bound to the antibody bound to the beads, and whatever proteins were bound to poly would then be pulled out as well. If we know what proteins we expect poly to bind to, or we want to see if poly is bound to, then we can use Western blots, also known as immunoblots, in which we can test for the presence of those different proteins, those different prey that we think might be there, using antibodies against each of those prey individually. This will only tell us, however, if the prey that we are asking for are there. Kind of like a game of Go Fish, if you don't ask the other player if they have a card, you're not going to know if they have a card. If we want to see their entire hand, we need a more unbiased approach, and so that's why you can use things like mass spectrometry or mass spec, where basically you're going to take the proteins and you're going to chop it up into little pieces, and then you're going to put those little pieces through a machine that's going to calculate the mass to charge ratio of those pieces, and then you compare that to a database, and you can figure out this peak with the pieces that are in there, and then you can take these little sequences that you generate and use them to figure out what proteins were actually present. That will tell you kind of what is there, not is it there. So that would be an IP mass spec, but more typically you're going to be using a co-immunoprecipitation, or most commonly you're going to actually be interpreting the results of a co-immunoprecipitation. And so we're going to talk about how you can interpret the results of these experiments to determine whether proteins are interacting with one another. Some things that you need to think critically about, as well as kind of how these experiments can be cool. So again, co-immunoprecipitation, as the immuno suggests, you're using antibodies. The antibodies can be directed directly to your antigen or to an epitope that you placed on your antigen. So it could be a flag tag or an HA tag that you added to the end of your protein. And this is going to allow you to use those same beads to capture a variety of different proteins. And then you can use the beads to pull out the protein and see what it's bound to. Often these days, you're going to be using these magnetic beads that make your life really simple. But you can also just kind of centrifuge the samples down to pellet out the beads that are attached to the antibodies that are attached to your bait. And then you can look for the presence of various prey proteins. And there are similar methods of pull downs where you use things other than antibodies. And so, for example, if you have a GST tagged protein, you can have glutathione linked resin that then you can use that to pull out the GST, which is going to pull out whatever bait you had that's bound to the GST. You can do similar things with strep tags and streptactin, with his tags and nickel. So various ways in which you can use other methods to pull out or precipitate, kind of remove from a solution, a protein, the bait protein, and then test to see what prey there are. And by having these other tags, that's also going to allow you to do things like purify it. And so you can make double use of those sorts of epitope tags and those sorts of tags. More on that elsewhere. Today, let's focus on interpreting the results of these experiments. Typically, you're going to see these Western blots, and so we're going to go over how to analyze these. So immunoblot is another term that you're going to see used often for a Western blot. and immunoblot is a little more generic. Sometimes on a gel, you might see like IB for immunoblot. That would be what it was talking about. So you might see WB for Western blot or IB for immunoblot. Speaking of a Western blot, let's think about what would happen if we did the Western blot without doing the pull down. So imagine we had the sample, and the sample contains this polyprotein, and it also contains these other proteins. And if we were to just do a Western blot detecting the presence of, say, this orange protein, we would see that the orange protein was present. And if we were to do a blot detecting with antibodies detecting the presence of the green protein, we would see that the green protein was present. And if we were detecting the polyprotein, we'd see poly was present. But that would tell us nothing about whether or not they were interacting with one another. So if we just did like a Comassi stain, if we did some sort of a gel and a total protein stain, we would see the bands for all these proteins, but we wouldn't know what they were. We could do a Western blot with antibodies against these different proteins to see whether or not the protein was present and kind of like how much of it was present. But we wouldn't be able to kind of know whether the proteins were interacting. In order to know whether they were interacting, then we would want to do something like a pull down. So now imagine that we do a pull down. So we pull down this polyprotein using antibodies directed against the polyprotein. We then wash off the proteins that weren't bound. And now if we take the proteins and run a Western blot just with the samples of the pull down, just the samples that were attached to the bead that was attached to our bait, well, now we're only going to see the presence of the proteins that were the bait or that were attached to the bait. Assuming, however, that we had specific binding. And so we don't want the protein to just kind of bind to the beads themselves. And so often you'll have a negative control where it's something like just the beads without your bait. And that'll kind of tell you whether or not the bands that you see are actually specific. Then, if you were to test for the presence of specific proteins, you should only see the proteins that were present because they were bound to the beads or they were bound to the protein that was bound to the beads. And so in this case, if we have that this protein interacts with the gray protein, we should see, and it interacts with the orange protein, we should see a band for the gray protein and a band for the orange protein. And we should see a band for our protein of interest, our bait, because, well, if the sample worked, you're going to have your bait present because that's what's going to happen when you pull it down is it should be there. Typically, when you're looking at a blot for a pull down, you're going to see an input lane, and then you'll see your immunoprecipitation or like your IP lanes or your pull down lanes. We'll look more detail at specific examples, but you'll see something like this where you have your input, and then you'll see like immunoprecipitation. It'll tell you what it's pulling out. It's important to include this input because that tells you whether or not the things were actually present in the sample and how much of it you're actually pulling out. So maybe you had very little of it in your actual sample, but you're pulling out a lot of it. Or maybe you have a lot of it in your original sample, but you're only pulling out a really little bit. So this will kind of tell you relative amounts. And it'll tell you if there was something there to actually be able to pull out. The strength of the bands in the input lane can also tell you about the relative expression in the sample. Just like any Western blot, the strength of the band is going to tell you about the quantity of the protein, but because different antibodies will have different sensitivities, it's not going to be able to allow you to compare different proteins to one another, just to compare the same protein in different samples, assuming that you loaded the same in all of the different wells, which is why you need to have some sort of loading control, such as your GAP DH or your actin or your tubulin to test for that. When you pull it out, you'll be able to test for the presence of various proteins. So in the sample, if you were to test for the presence of your bait, you should see a band for the bait. And this should always be the case because that's going to tell you that kind of your strategy is working. Remember, your beads are going to pull out your bait, and if you don't pull out your bait, you have no hope of pulling out things that were attached to your bait. Then you can probe for the different things that you're interested in that are attached to your bait. So if you were to probe in different strips for each of these different proteins, you would only see a band corresponding to this little gray protein and this little orange protein. Or at least you should only see a band because those are the only ones interacting with the bait in the sample. If you see other bands, then that could be a problem. But those bands that were binding non-specifically to the beads should be showing up in a negative control lane. So typically you'll have some sort of negative control where maybe you just have the beads themselves. And sometimes you might see multiple negative controls where you might just have like the tag. If you put a tag on your protein, you might just have the tag itself to see whether the tag itself is binding to anything. Things like this, various negative controls to make sure that the proteins that you do see in your pull down are there because they were bound specifically to your bait. When you're looking at a co-IP, often you're going to see a kind of table looking thing. The table above the results will tell you about the different experimental samples. So these might be different cell types. They might be the same cell type with or without various chemical additions. They might be the same cell type over time. But you'll have kind of two big kind of broad columns, the input and the IP. So each of these columns is going to represent a different sample. And above the sample, it's going to tell us kind of what is in the sample. A plus sign is going to tell you that something is present, and a minus sign is going to tell you that thing is not present. And then you should have each of these kind of columns should be repeated in the immunoprecipitation. Often with the input, you're actually going to be loading a very small amount compared to how much you load with the IP because there's so much protein present, and only a small portion of it's actually going to get pulled out by your beads. But you'll have your input, and here should be everything that's present. And then if it's important to have this input so you know that if it was present in order to see whether or not it was actually pulled out. Because if it wasn't present, then there's no hope that you would pull it out. And therefore, not seeing it pulled out isn't telling you whether or not it can interact with the protein. It just tells you that it wasn't present in the sample to interact with your bait. In the table, what you'll see is you might have different things that were tested. So in some samples, say you had an HA tagged protein A, and in some samples, you had an HA tagged protein B. Then in this example, you were immunoprecipitating for the HA tag. If the tag was attached to protein A, well, now what you would have is you'd be pulling out protein A, so protein A would be your bait. If your tag was attached to protein B, well, now if you pull out the tag, protein B would be your bait. You could also imagine that you could get the same results if you had an antibody that directly recognized protein A or an antibody that directly recognized protein B. But having this HA tag on it allows us to use the same antibody to recognize both protein A and protein B, but separately, or else it wouldn't be that useful. If we go row by row, we can kind of see, okay, so protein A and protein B. If we don't have either of them, and we're doing an immunoprecipitation for that tag, well, then we shouldn't see anything present in the immunoprecipitation because we don't have an HA tagged thing in order to pull out. So that would be an example of a negative control, and we wouldn't want to see any bands in our co-IP, in our immunoprecipitation lane. Now, if we do an IP, but we have HA tagged protein A, now our HA tagged protein A is going to be our bait. If we pull it out, we should see it. And we should also see that we have whatever it's bound to. And so in this case, we could see that it would be bound to protein C, and it would be bound to protein D, but it would not be bound to protein E. This doesn't, however, tell us that our protein doesn't interact with protein E, because we can see in the input that protein E wasn't even present in the sample to be able to interact with. So from this results, we can say that it looks like protein A is interacting with protein C and protein D. Now let's imagine we do the same thing with protein B. So instead of having HA tagged protein A, we have an HA tagged protein B. We can see that we're still going to see a band for this protein B, this HA tagged protein B. This is going to be our bait. And so note too that if we're doing a blot against the HA tagged tag, here we see these bands are different sizes for our HA protein A and our HA protein B because they're different proteins, but they're both attached to the same tag, and so they're both able to be recognized by that same antibody. In this case, what we see here is that we don't see a band for protein C, but we do see a band for protein D. So we can say that protein D appears to be interacting with protein A. And protein C, well, can we tell if it's interacting? Well, if we see it's in our input, but it's not in the pull down, so it does not seem to be interacting. This is different from protein E. Protein E we didn't see in our input, so we can't tell if it's interacting. Well, I mean, it's not interacting in the sample, but we don't know whether it could interact. It's just not present, so we can't really say much about it. But protein C, we know that it was present, but we know that it's not being pulled down, so it doesn't seem to be interacting. Note that this is kind of inconclusive still, no matter what you see, because depending on how strong the interaction is, you may not see it. The interaction might be transient. It might be washed off by the time you measure it. It might be that they only interact under certain circumstances, maybe only when there are certain other things in the cell present, maybe there's indirect binding. So that's another thing, like you don't know whether the binding of your prey is directly to the bait or if it's actually happening through another protein that's bound to your bait that you don't see because you're not testing it. So lots of complications, but at this most basic level, we could do things like say, okay, from these results, it looks like protein A is interacting with protein C and protein D, and protein B is interacting with protein D, but not protein C. Note too that here we still want to have a loading control. So we want to have something like our gap DH so that we can compare between the samples so that if we were to add a lot more of one of the samples, we'd be able to normalize to it so that we would know that it's not that there's more of this protein being made in these cells, it's just that we loaded more into the well. Now let's look at a real example. In this paper, these scientists were studying these glutamate receptors, these AMPR receptors, and whether they were interacting with this protein, PRRT1. Don't need to worry about what those proteins actually are, but let's worry about how we can interpret the results of their precipitations. So in this case, we have either a flag tagged GluA1 or an HA tagged PRRT1. Often scientists will do this where instead of, say, having one HA protein A and then one HA protein B and have those be different samples, you can have different tags on different proteins within the same sample. And then you can do immunoprecipitations against either of the proteins. This serves as a good sort of like control. So if you pull out one protein, do you see the other? And if you pull out the other protein, do you see the first one? In this case, what they're doing is they're doing an immunoprecipitation against the HA tag. We can see that the HA tag is attached to their PRRT1. So our HA PRRT1 is going to serve as our bait. And then we're going to be looking for the GluA1 as our prey. We're going to be detecting GluA1 by detecting the flag tag that's attached to. So this would be our IB, our immunoblot for the flag, and our IB, our immunobat for the HA. Sometimes you won't see IB here, you'll just see like anti, or sometimes you'll just see just flag or HA. It all depends. You'll see different formats on different papers, but what you're looking at in each of these kind of strips is going to be the probing for a specific protein. In this case, they're probing for the flag tag, which is going to tell you the presence of GluA1. And in this row, they're probing for HA, which is attached to PRRT1. So it's going to indirectly tell you about the presence of PRRT1. If PRRT1 is present in the input, you should see it over here. So we can see in this first sample, PRRT1 was not present, and so we don't see a band. But GluA1 was present, so we do see a band. In the second sample, both of them were present, so we see bands for both of them. So that was in our input. Now let's look at the immunoprecipitation. In this case, we're immunoprecipitating the HA tag. We saw in the sample that we don't have the HA PRRT1, and so we also saw that if we pulled out, if we had antibody against HA, we didn't see anything, so there's nothing else HA tagged in these cells. And so if we were to pull out HA tags, we wouldn't see anything. We shouldn't expect to see either the flag tag GluA1 or an HA tag PRRT1 because they weren't present in the sample. So this would be an example of a negative control. Then we have our actual sample where we have these two proteins present. We know that if we do the immunoprecipitation against the HA, so this would be our bait, we should see our bait. And indeed, we do see our bait. This is telling us that our beads, our strategy is kind of working. Now we want to see, okay, well, is it interacting with GluA1? In order to do that, we need to look at the row that corresponds to the GluA1. Remember, in this example, the GluA1 was attached to the flag tag. So by probing for the flag tag, we can probe for the GluA1. We see a band for the flag tag, which tells us that GluA1 is interacting with your HA tagged PRRT1. So from the results of this experiment, we can at least kind of tentatively say that GluA1 interacts with PRRT1. But what about GluA2? They do a very similar thing and you see very similar results. So we have our input. We see here that in this first lane, we have flag tag GLUA2 present, but not the HA tag PRT1. Because we're immunoprecipitating the HA tag, the HA PRT1 would be our bait. But our bait is not present in the sample, and so we shouldn't see it present over here. We can see that we do see some nonspecific staining over here, which might indicate that our antibody is not that specific, and so we need to be careful that we don't go and quantify this when that's really not corresponding to the bands. In order to see like the real interesting results, we need to look at the lane that has both of the things that we're testing it for, that has both the bait and the prey, because only then can we tell whether they were interacting. And in this example, it seems like they were interacting because when we pulled out our bait, we pulled out the HAPRRT1, we saw that we also pulled down the flag tagged GLUA2. But what if we test GLUA3 and GLUA4? With GLUA3, we see similar results. We also see similar results with GLUA4. However, we can tell we kind of get more of this kind of either unspecific like antibody binding or unspecific binding to the bead. So that's something that you want to keep an eye out for and be a little cautious of. If you do see a lot of nonspecific binding, you can do things like raise the salt concentrations. The salt's going to kind of compete for the binding to the beads, and therefore it's going only the more specific interactions, the stronger interactions can survive. So it looks like all of these GLUA4, 3, 2, and 1 can all interact with the PRRT1. What about PRRT2? Here, we see PRRT2 is going to be our bait, and our prey is going to be GLUA1 in this case. What we'll see is that here, well, we have a very little of it that gets pulled out along with when we pull down our PRRT2. So it seems like GLUA1 is not interacting with PRRT2, at least not nearly as well as it interacted with PRRT1. In addition to having the negative control where they don't have a thing with an HA tag, in order to make sure that nothing is binding directly to the beads, sometimes you'll also see a kind of non-specific protein used. And when I say like non-specific, it's something that like that your protein shouldn't bind to, or there's no evidence that has previously been found that it'll bind to. And that'll tell you, okay, well, is this kind of binding more specific, or is it kind of a generic blobbing on binding? And so in this case, they have flagged NRXN1. So in this case, their HA tag is still the PRRT1. And so our PRRT1 is going to be our bait. NRXN1 is the prey that they're testing for. When they pull out their bait, PRRT1, they see that they do not pull down any NRXN1. But they know that their antibody for NRXN1 worked and that the NRXN1 was present in the sample because they see it in their input, but they don't see it in their pull down. And so that's going to tell you that that the PRRT1 does not seem to be interacting with the NRXN1, which tells us you can be a little more confident about the results that the PRRT1 is interacting with these GLUA receptors. So no matter how complicated the immunoblotting or Western blotting results from a co-IP or pull down assay look, you really just need to play a game of spot the differences. First, you want to identify your bait. Your bait is going to be the thing that you're directly pulling out. Often to actually find out what the bait is, you'll go and you'll look and you'll see something like IP, or you'll see something like pull down, something in kind of the right side of the blot, where it's going to indicate what is actually being pulled out by the beads. The thing that gets directly pulled out is your bait. In this case, we have our bait B. It's pulling out this HA, and this HA is this tag that's going to be attached to protein A or protein B. You can also imagine having an immunoprecipitation against just protein A itself or protein B itself, but by using these epitope tags, we're able to stick the same tag on multiple proteins and use the same beads to pull out multiple proteins, one at a time. And so in this case, our bait would be the HA. To see if the bait was present in our sample, we look at the input. To see whether our beads were working, we look and see that we actually, in our antibodies against that were working. We look and we see in our pull down, we look at the lane where we're doing a Western blot, or we're doing a blot, we're using antibodies that then detect the presence of the bait. We should see this in our input to know that it was there to pull out, and we should see it in our pulled out to know that our pulling out was actually working. But what's more interesting is figuring out what the bait was bound to. The things that the bait were bound to are called your prey. If you know what prey you want to look for, you can use antibodies against each of the prey individually. If the prey was present in the input, you should see it on the input side. This will tell you that it was there to interact with. It'll also tell you that your antibodies were working for it. If then you don't see it in your pull down, it's telling you it's not interacting. If it wasn't present in your input and you don't see it in your pull down, it doesn't really tell you anything because it wasn't there to begin with. So in this example, we're pulling out the HA tag protein A, so that HA tag protein A would be our bait. Similarly, in the case where we have the HA tag on the B, and then we're pulling out based on that tag, the HA tag protein B would be our bait. Assuming that nothing is interacting indirectly with the tag itself, it's therefore basically protein A and protein B that would be your bait in those two scenarios. And how do you know what's in your samples? Remember, that's where you go and you look above the columns where you see this kind of table telling you about what's what. So go column by column, see what's in the sample, see what bands are there in the input, and then see whether those bands are still present in the pull down, telling you that they were pulled out in your pull down. You can use different antibodies to detect the presence of different proteins, and you can use different baits to detect the presence of different interactions. Often what you'll see is then you'll see the reversed reactions. So maybe in this example, you had a flag tag glue A1, and you're looking for an HA tag PRT1. In this case, you did an immunoprecipitation for the HA tag, but you could also imagine doing an immunoprecipitation for the flag tag. You could then use the same antibodies, and you should get the same results if the binding was specific and it was strong enough. You also want to see some sort of negative control to make sure that the prey wasn't directly binding to the beads, to make sure that your antibodies are kind of specific, and that would be kind of some sort of negative control. If you don't watch your samples off well enough, you might see some bands here. If you have some non-specific binding to the beads, you might see some bands here, various things like that. You don't want to see bands in your negative control. You also want to see some sort of loading control. For a housekeeping protein, something that's ubiquitously expressed, something like GAPDH or actin or tubulin, this is going to tell you that you loaded similar amounts in each of these different lanes, at least over here. It gets a little complicated then when you try to do the immunoprecipitation, because you can't then have the housekeeping gene unless you don't want to see it in the pull down, because that would mean it was kind of non-specifically interacting. But you would hope to at least see it in your input. And then you can do fancy ways of normalizing your data. But you want to see that if this band were to be darker than this band, it was because you had more of this band present, not because you just loaded more of this total sample present. If you loaded more of this total sample present, however, you would have more of the GAPDH present, and then you'd be able to normalize between them. So no matter how complicated these figures might look, remember, take a deep breath. You've got this. Just look at the columns, look at the rows, go step by step. Think logically, maybe block out lanes with a piece of paper or something on your screen to make it so that you're focusing on one thing at a time. Get practice. Practice makes perfect, or at least practice makes easier. So you got this, you got this, you got this.